And we have Jaak Panksepp, who will introduce our first speaker.
Thank you.
Good morning. It's a real pleasure to introduce J Suomi. He needs no introduction, but he carries on the grand tradition that Harry Harlow started after having the deep insight on how to approach social attachment, social bonding, the deep mechanisms that make us human eventually. And he was about to move to the University of Texas 25 years ago, and the National Institute of Child Health and Human Development decided to make him an offer he could not refuse. And for 25 years on the Poolesville campus, he has developed a superb primate research facility where the output gets better every year. Steve, it's a delight to invite you to the podium.
Can everybody hear me? First of all, Jaak, thanks so much. You're a hero to me. It's been for a long time. It's a real pleasure to be back in South Bend and on the Notre Dame campus, and particularly to be a part of this wonderful symposium and have the opportunity to tell you about some of the work my colleagues and I have been doing in my laboratory at NIH over the last few years. As Jaak alluded to, my laboratory is not on the main campus in Bethesda, but rather out in the countryside, near about 30 miles up the Potomac from downtown D.C. and 28.6 miles from the main NIH campus. It's near a little town called Poolesville, so when I talk about the Poolesville colony, that's what I'm referring to. And out there in the countryside, we have the kind of space available to have a colony of approximately 400 rhesus monkeys, many of them living in naturalistic settings, or EAA settings, as the term for this meeting is. And since its inception, my lab has had a major focus on the study of individual differences in temperament or personality and the biological substrates that underlie these individual differences, and how genetic and environmental factors act, and we now know, much more importantly, actually interact to shape individual developmental trajectories. So, for example, over the years, we've been especially interested in a couple of subgroups of monkeys in our colony, and also subgroups that we've been able to observe at 2 field sites that we've been fortunate to have access to over the years. The first subgroup comprising about 20% of the population of monkeys out at Poolesville, as well as 20% of the monkeys at these 2 field sites, seem to be unusually fearful or anxious in the face of novel or mildly challenging circumstances and stimuli. So stimuli and circumstances that other monkeys find really interesting and will readily explore, these monkeys try to avoid. And if they're forced to confront these situations, they show profound arousal of a variety of biological systems. The systems we spent the most time studying has been the HPA axis. These monkeys consistently have elevated levels of cortisol for longer periods of time under comparable circumstances than others in the group, whether you're measuring the cortisol in blood or in saliva or more recently in hair. These monkeys in social situations look to be relatively shy, and they look very much like a subgroup of children that people like Jerome Kagan and Nathan Fox have been studying for the last quarter century, children who share the behavioral and biological propensities, tendencies of this subgroup of monkeys, who we call our uptight monkeys, and children who have been called behaviorally inhibited and who we now know are at risk for developing childhood anxiety and depressive disorders, and for some of whom this risk carries on into adolescence and even into adulthood. There is a second subgroup of monkeys that we've also been interested in. This subgroup comprises maybe 5 to 10% of the population out at Poolesville, and again 5 to 10% of the monkeys that we've been watching in nature. These monkeys seem to be unusually impulsive, and their impulsivity gets them into trouble. They do stupid things that other monkeys know better than to try, like get between a high-ranking adult female and her offspring, or they may repeatedly confront a dominant adult male. And when that male beats them back, they're right back in that male's face. And when that male beats them back again, they're right back in that male's face. So not surprisingly, they tend to elicit a lot of aggression from others in their group. And they themselves are often aggressive, often inappropriately aggressive, and more than that, once they get into an aggressive encounter, whereas most monkeys know how to back off when things start getting hot before things get out of hand, when these monkeys are involved in an aggressive encounter, often the intensity of the aggression escalates to the point where somebody can actually get hurt. And these monkeys also have biological characteristics that seem to differentiate them from others in their social group. The characteristic we've paid the most attention to over the years is an apparent deficit in serotonin metabolism that we see in terms of chronically low levels of the primary central serotonin metabolite in cerebral spinal fluid, 5-hydroxyindoleacetic acid, or 5-HIAA. And similar to what's been found in very aggressive humans, monkeys who tend to be impulsively and explosively aggressive like this chronically have chronically low levels of CSF5-HIAA. Now, these monkeys, whether they are aggressive or fearful or shy or relatively normal, in the wild live in complex social groups. And let me tell you a bit about the rhesus monkeys as a species before we go into some of the details. And then I'll describe some of the effects of early experience that we've been able to see, characterize some gene-by-environment interactions, and then focus on some recent work we've spent the last few years looking at very early mother-infant interactions and what they are telling us about very early aspects of development. Rhesus monkeys are not our closest relatives. They only share about 95% of the genes that we have. By contrast, as Jacques told us last night, bonobos, our closest relatives, share between 98 and 99% of our genes. But rhesus monkeys are a lot more like us than our bonobos or chimpanzees or any of the other great apes in one fundamental respect. They are a success story. They are the world's second most successful species of primates after us. There are more rhesus monkeys living in the wild, encompassing a greater geographic range, experiencing a wider range of climates than any other species of non-human primates. And in the Indian subcontinent, you can find them everywhere from rainforests to hardwood forests to coniferous forests to savanna areas to the edges of the desert, and even in the foothills of the Himalayas where the winters are a lot more severe than anything around here. So we have no problem maintaining a colony of rhesus monkeys in outdoor settings in rural Maryland with minimal shelter. And furthermore, they're one of the few species of primates, we're one of the other ones, that can be genuinely called a weed species. That is, if you take them out of their natural habitat or their initial habitat and put them someplace else, they almost always thrive and expand their populations, whereas that is not true for most other primate species, which even in their own habitats are severely endangered or threatened. So every time somebody's taken rhesus monkeys out of India and put them someplace else, whether it's an island off the coast of Puerto Rico or the swampland outside of New Orleans or the high arid plains in western Texas or the Sacramento Valley in California, where it gets to be over 100 degrees every day during the summer, or rural Maryland, in all these places, these monkeys have thrived. And finally, as a species, they're unbelievably resilient to all sorts of insults, both man-made and natural. I can tell you lots of stories. I'll leave you with one very brief one. All of you remember, I trust, Hurricane Katrina, which trashed the Mississippi Gulf Coast in New Orleans 5 summers ago. What you may not know is 30 miles north of New Orleans, on the northern shore of Lake Pontchartrain, is the Tulane National Primate Research Center, where they had 3,000 monkeys living in outdoor pens when the hurricane hit. And when the hurricane passed over, it blew away all those fences, all those pens. Yet every single monkey survived. They didn't lose any one of that 3,000. And we think we know how it happened. In each of those pens, the animal caregivers had put large concrete culverts, the sort of thing that they use for drainage under expressways, so that the monkeys would have a place to get out of the hot Louisiana sun. And almost certainly what happened when the hurricane hit is these monkeys all headed for those culverts, hunkered down, rode out the storm, and then waited for the humans to show up with their breakfast in the next couple of days. So these monkeys survived the Hurricane Katrina a lot better than most humans did. In the wild and in those captive environments that so provide, such as what we've got out at Poolesville, these monkeys live in large social groups called troops. The troops can range in size from 30 or 40 individuals on the small end to up to several hundred on the large end. But whether the troops are large or small, they all have the same basic social structure. Every troop is organized around several female-headed, multi-generational families, or matrilines, plus males who have come in from the outside. And this arrangement derives from the fact that rhesus monkey females stay in the troop in which they're born for their entire life, whereas rhesus monkey males stay only until puberty, and then they leave. They either leave voluntarily or they're physically kicked out of the troop, usually by unrelated females. These males then typically join all-male gangs, hang out in the gangs for anywhere from several months to a year, and then try to work their way into a new troop. So every troop has plenty of adult males and females in it, but the females have all been there since birth, and they're all related to one another, at least within their respective families, whereas the males have been there only since puberty, and initially they're related to no one until they start having offspring of their own. And here's one typical rhesus monkey family. Here's the mother, newborn infant, grandmother's back there. This is an older sister of the infant. This is a younger sister of the mother. The other thing you should know about rhesus monkey social organization is within every troop there are multiple dominance hierarchies. There's a hierarchy between families, such that every member of the highest-ranking family, including infants, outranks everybody in the next highest family, including adults, who outranks everybody in the third family, etc. There's a hierarchy within each family that follows the basic rule, younger daughters outrank older daughters. And it's easy to see how this starts, because a mother will preferentially defend her newborn daughter from the harassment of a jealous older sister. So even though the older sister is bigger and stronger than her younger sister, with the mother to back her up, that younger sister can get away with anything she wants in those interactions. The interesting thing is that this arrangement is maintained as long as both sisters are still alive, even after the mother is no longer around to differentially reinforce that, because where the mother leaves off, other monkeys take over and continue to give deference to the younger sister in her interactions with their older sibling. There's a hierarchy among the males that come into a troop that's roughly related to tenure. That is, the longer a male is in a troop, the more likely that male is to be high-ranking. But of course, it's not tenure or length of time that's important. What's really important is how good that male is at making friends and sustaining alliances, not only with other males, but more importantly with high-ranking females who carry the real clout in the troop. And males that are good at that stick around and move up, and males that are not so good at it give up, leave, and seek their fortune elsewhere. And then finally, there's a hierarchy among the infants that are born into a troop every year that basically reflects the position in the troop of their mother. So it's not that some infants are bigger or stronger or faster or quicker or smarter than other infants. Those differences undoubtedly exist. It's that other monkeys associate a particular infant with a particular mother and afford it equivalent social status. So life in a rhesus monkey group is a pretty complicated business, and for individuals to survive in such a complex group, let alone thrive, they have to not only acquire a complicated behavioral repertoire, but they also have to acquire a great deal of knowledge about the troop. Who's part of the troop and who's not, who's related to whom, where does each individual fit in these multiple dominance hierarchies, as well as recent social history, who's been getting into fights recently, who can you count on to back you up if you get into a fight yourself. And monkeys that do this, learn this, and follow the rules inherent in these complex dominance hierarchies do very well in this complicated social situation, and monkeys who are either unable or unwilling to follow these rules don't last very long at all. So a few words about the socialization process. Rhesus monkey infants, who, by the way, grow up about 4 times faster than we do, which is an enormous advantage for those of us who are interested in studying the long-term effects of early experience or lifespan development or the transmission of characteristics from one generation to the next. We can see a generation in 4 or 5 years instead of having to wait 15 or 20 for the human equivalent. So when I give you an age in rhesus monkey months or years, multiply by 4, and you'll get a rough human equivalent. At any rate, rhesus monkey infants spend virtually all of their first month of life in intimate social contact, physical ventral-ventral contact, or no more than arm's length away from their mother. And during this time, a strong and enduring attachment bond is established between mother and infant. It's a bond that's functionally equivalent to the attachment bond that human infants routinely form with their caregiver. And it's an historical fact that Bowlby's attachment theory, the biological basis of Bowlby's attachment theory, actually comes from studies of rhesus monkey mother-infant interactions. Bowlby was a good friend of Robert Hinde, the British ethologist, from Cambridge, who at the time Bowlby was developing his attachment theory, was just shifting his area of interest from study of song learning in birds to the study of mother-infant interactions in rhesus monkeys. And Hind introduced Bowlby to my mentor, Harry Harlow, who at that very same time was doing his classic studies of cloth and wire artificial surrogates, studies that still show up in introductory psych books 50 years later. And between Harlow and Hind, Bowlby had all the ammunition he needed to posit a biological basis for attachment that had evolutionary roots, that was a product of evolution rather than something simply involving effective phenomena. So during this month, while that attachment is being formed, mothers are very protective of their offspring. They're very picky about who they let interact with their offspring. They don't let adult males get too close, which is one reason why in this species, not necessarily in other primate species, but in this species, fathers have almost nothing to do with the early caregiving of the infant. These females usually don't let unrelated females get too close. In this particular, mother's not going to let the photographer get one step closer. These infants don't stay infants forever, however, and beginning in the second month of life, they start leaving their mothers for short periods of time to begin exploring their environment, and consistent with Bowlby's attachment theory, they'll use their mother as a secure base. That is, they'll go away from the mother for a few seconds and a few feet, and then go running back to the mother for a period of contact, and go explore a little bit more, back to the mother for a period of contact. It's almost as if the infant were attached to the mother by a long rubber band. And as the weeks and months go by, these exploratory forays become more frequent, involve greater distances and amounts of time away from the mother, so that by 6 months of age, these youngsters are spending only about 1/5 of their waking hours in actual physical contact with their mother, as opposed to the 100% that they were doing in that first month. Yet the mother's physical presence in the immediate environment is absolutely crucial to sustain this early exploration, because if you lose your secure base or access to your secure base, any motivation to explore will disappear rapidly, and you'll become pretty upset emotionally. So even though infants aren't actually interacting physically with their mother that much in these later months, their mother is still a crucial part of their social world. Who these infants do interact with during this time are peers, other monkeys about the same age as themselves. And this is not by accident. In nature, rhesus monkeys are seasonal breeders. They copulate year-round, but the conceptions only occur within a 2 to 3 month window every year, which means 80 or 90% of a troop's infants in 1 year are born within a 2 to 3 month window. And what this guarantees is that a youngster born during that time will have plenty of other youngsters who are about the same age and who have about the same physical, cognitive, and social capabilities as themselves. And as they're growing up, this is who they choose to hang out with. So throughout the childhood years, the rest of the first year of life, the second year of life, and the third year of life, these monkeys spend several hours every day in active social play with one another. And it's in the context of this play that virtually every behavior pattern that's going to be crucial for normal adult functioning can be developed and practiced and perfected long before it actually has to become functional. This is especially true for reproductive behavior, and it's just as true for the socialization of aggression that comes into these monkeys' repertory between 4 and 6 months of age. And work by Harlow and his colleagues way back in the 1960s, as well as much more recent work in the last decade by people in the Netherlands, have shown that rhesus monkeys who are otherwise well socialized but denied the opportunity to play with peers while they are growing up inevitably grow up to have real problems with reproduction and real problems with aggression. Now, not all of our monkeys out at Poolesville are raised this way. Some of them are cross-fostered to unrelated females at birth, so we can disentangle genetic and environmental factors, and others are reared without access to parents at all. This is a rearing procedure that Harry Harlow developed in the late 1960s and has been used extensively at other laboratories around the country, if not the world. This is called peer-only or together-together rearing. Here, infants are separated from their biological mother at birth, well before any attachment can start developing. They are hand-reared in a neonatal nursery for the first month of life, and during that time, later in that month, they are put in with other monkeys like themselves who are likewise peer-reared, and these monkeys live together 24 hours a day, 7 days a week for the first 6 months of life, at which point they are moved into larger social groups that also contain their mother-reared counterparts. This is the age in which weaning normally occurs in the wild. So all of the differential early experience I'm going to be telling you about is only for the first 6 months of life. Thereafter, mother-reared peer-reared and their mother-reared counterparts are all living in the same physical and social environments. And when monkeys are reared this way, they develop hyper-attachments to one another, extraordinarily strong attachments, but these attachment bonds, while very strong, are actually dysfunctional. Because what happens is these youngsters spend excessive amounts of time clinging to one another instead of going out and starting to explore their environment. And when you think about it, a peer is not nearly as good at providing a secure base as even a relatively poor mother. So these monkeys don't spend a lot of time initially becoming familiar with their immediate environment. And when they get to the age where they should be starting to develop the play, they do develop patterns of play, but the level and the complexity of play and the intensity never reaches the level that you see in their mother-reared counterparts. And when you think about it, it's not surprising. These monkeys have to be playmates and attachment objects at the same time, and they don't do a very good job of either. So these monkeys, even though they're with potential playmates 24 hours a day, are actually play-deprived as they are growing up. And this rearing has its consequences. For one thing, peer-reared monkeys, as they get older, start looking more fearful and anxious in new situations than you would expect had they been reared by their mothers. They look very much like that 20% of the naturally occurring 20% of fearful monkeys that occurs in nature. And like that 20% in nature, they show excessive HPA activity, as evidenced by the significantly higher levels of cortisol, at least through the first 2 years of life. Later, these seem to normalize, and I may add, many of the rearing condition effects I'll be describing actually are reversible, but that's another talk. So these monkeys, behaviorally and biologically, look very much like the naturally occurring 20% of fearful monkeys in the wild. But they also look like the 5 to 10% of naturally occurring impulsive, aggressive monkeys, because as these monkeys get a little bit older, they start showing and developing these explosive patterns of aggression, escalating aggression. And like those monkeys, 5 to 10% in the wild, they also show deficits in serotonin metabolism. Whether you're looking at the first year of life when these levels are relatively high, there's a significant rearing condition effect, or later on during development when the levels have dropped for everybody, but if anything, the rearing condition difference is larger than before. And this is not the product of genetic differences, even though we now know that differences in serotonin metabolite within a rearing group are highly heritable. That is, there is a significant genetic component. The same is true of cortisol. So even though there are these individual differences in cortisol and 5-HIAA, in nature have a significant hereditary component, they also can clearly be altered by early experience. When these monkeys get to be juvenile adolescents and young adults, some of my colleagues in the Alcohol Institute get their hands on them and run them through the monkey version of a happy hour, where over a period of weeks and sometimes months, for once a day, for an hour a day, these monkeys have unlimited access to 9% NutraSweet flavored alcohol beverage or non-alcoholic NutraSweet flavored beverage or plain tap water. Under these circumstances, some monkeys consume more alcohol than do others, and as a group, the peer-reared monkeys consistently consume significantly more alcohol than their mother-reared counterparts. Recently, we've been able to do some neuroimaging studies. Jacques referred to one of them yesterday. These are PET scans and structural MRIs of differentially reared monkeys. Here are data from the paper by Akisi et al. That Jacques cited yesterday. These on the right, on the left is serotonin binding potential. On the right is cerebral blood flow. These are PET scans taken when the monkeys are 2 years of age. That is, they were differentially reared for the first 6 months, but for the last 18 months, they've been living in the same social group. And the comparisons are between a front view of mother-reared composite versus peer-reared, top view of mother-reared composite versus peer-reared, side view of a mother-reared composite versus peer-reared, and similarly for cerebral blood flow, mother versus peer, mother versus peer, mother versus peer. And simple inspection shows you that the peer-reared monkeys' brains aren't lighting up as much as their mother-reared counterparts. And I'm sorry, Alan, this is not broken down by laterality, but as you pointed out, there is a laterality difference, but across the entire brain, whether you're looking at the raphe, the thalamus, the striatum, the temporal, or the frontal regions, frontal cortices, peer-reared monkeys have significantly less cerebral serotonin binding potential than their mother-reared counterparts, and the same is true for cerebral blood flow. We recently published, it's still in press, a paper looking at serotonin 1A receptors. We see the same kind of rearing condition difference. We recently published a structural MRI paper looking again at 3-year-old mother versus peer-reared monkeys, and their brains are structurally different. So peer rearing has its effect not only at the level of behavioral expression and emotional regulation, more fearfulness, more aggressiveness, at the level of neurohormonal output, higher levels of cortisol, at the level of neurotransmitter metabolism, lower levels of CSF, 5-HIAA, even at the level of brain structure and function. And very recently, we started to look at gene expression. Here, we've been collaborating not only with my ex-graduates, ex-postdoc now at Wake Forest, Alison Bennett, but more importantly with Michael Meany and Moshe Ziff at McGill University. And Moshe Ziff now is able to do genome-wide evaluations of methylation status for every gene in the genome from various forms of tissue. And our first crack at this has been to look at tissue obtained from 8-year-old, that is fully adult, rhesus monkeys who either were peer-reared or mother-reared for the first 6 months of life and thereafter living in the same situation. And what you're seeing is genome-wide scans of methylation patterns here, and the results have been astonishing. Fully 1/5 of the entire rhesus monkey genome, over 4,400 genes, are differentially methylated as a function of what happened in that first 6 months of life. So here is just as an example, 2 chromosomes, chromosome 1 and chromosome 2. Top line is prefrontal cortex of data. Second line is lymphocytes from white blood cells. Each bar is a gene that's significantly methylated. If it's above the line, it's more methylated in mother-reared. If it's below the line, it's more methylated in peer-reared, both in prefrontal cortex and in lymphocytes. Here's chromosome 2. Here's an example. Here's chromosome X. Here, interestingly enough, at least in prefrontal cortex, most of the methylation patterns are over-methylation in mother-reared monkeys. So, moreover, there's some correspondence between some of the methylation patterns you see in brain and in blood. So, for example, on chromosome 1 for these particular genes, if a gene is overly methylated in a mother-reared monkey in brain, it's also over-methylated in mother-reared monkeys in blood. Similarly, if it's over-methylated in peer-reared monkeys in brain, over here, this thing isn't very good, it's over-methylated in peer-reared monkeys in blood as well. Here are examples. It's about 30% of those 4,400 genes follow this pattern. About 25% follow the opposite pattern. That is, if it's over-methylated, genes that are over-methylated in brain for mother-reared tend to be over-methylated in blood for peer-reared. 25% of that. Between the two of those, you've got 55% of these 4,400 genes for which information on particular genes in blood can give you information on particular genes in brain. And the reason this is important is while current technology has only let us get brain samples once, you can get blood samples every week, and that's what we're doing right now. We're doing a prospective longitudinal study, getting blood samples every week and watching these rearing condition differences emerge across the genome as these monkeys are growing up. And furthermore, they're not random. Here, for example, are the genes associated that are differentially methylated that are in the HPA pathway. So the HPA pathway has multiple genes in it. These particular genes are differentially methylated as a function of early experience. And you can see some of them are showing up both in brain and in blood. Some, the vasopressin genes only in brain, many of the immune system genes only in blood, and some, again, in both. This is methylation patterns. More recently, we're also looking now in a collaboration with Steve Cole at UCLA and the previously mentioned James Heckman, the economist from the University of Chicago, looking at actual gene expression using microarrays looking at leukocytes. And here, our first set of data come from 4-month-old monkeys, and they too are astonishing. These are heat plots. Each of these vertical lines is a DNA point across the genome, genome-wide. If it's red, it's overexpressed. If it's green, it's underexpressed. Across the top, each horizontal thing is an individual monkey. So you have mother-reared, 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 peer-reared, 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 and there's almost no overlap. if it's red, if it's overlap methylated, if it's overexpressed in mother-reared, it's underexpressed in peer-reared. If it's overexpressed in peer-reared, it's underexpressed in mother-reared, with one exception, MR55. Mother-reared actually looks more like a peer-reared monkey. MR55 is the offspring of a primiparous mother who rejected it and neglected it. So work from my colleagues in Atlanta have shown that they have a colony of monkeys where some families of mothers, generation after generation, neglect and abuse their offspring. The offspring look very much like peer-reared monkeys, more fearful, more aggressive, higher cortisol, lower 5-HIAA, and we're seeing it in the genome. So these are main effects of early environment. What about gene-by-environment interactions? Well, we've taken a candidate gene approach over the last decade or so, looking at individual genes for which humans and rhesus monkeys have the same gene, and furthermore have the same polymorphism. The gene that's gotten the most attention is the serotonin transporter gene. This is a gene thought to regulate the reuptake of serotonin presynaptically. It's the same area that drugs like Prozac seem to work. And in both humans and non-human and rhesus monkeys, just rhesus monkeys, there's a polymorphism due to length variation in the number of base pairs in the promoter region. And these are functional differences in that in both humans and in monkeys, the short allele, the short version of the gene, is associated with less efficient RNA transcription from DNA. I won't go in, I don't have time because we're running a little late to go into the human literature, but some people have reported that carrying the short allele makes individuals disproportionately likely to be incarcerated for severe aggression, to be hospitalized for severe depression, or to be suicide victims. Others have not been able to replicate that finding. Probably some of you know of the Caspi and Moffitt now landmark study of 2003 in Science, where in Dunedin, New Zealand, humans in their mid-20s who had the short allele had higher levels of depression and more depressive symptoms than those with the long allele, but only if they also had either high levels of concurrent stress or a history of child maltreatment and or abuse. Absent that, those environmental characteristics, having the short allele confirmed no greater risk for depression than having the long allele. Caspi et al. Called this a gene-by-environment interaction. But we've been able to characterize all the monkeys in our colony, and some have the short allele, and some have the long allele, and some were nursery-reared, peer-reared, and some were mother-reared. The question is, does it make any difference? And we found the answer to be interesting. Let's look at serotonin metabolism. Here, if you're peer-reared and you have the short allele, you have a deficit. If you have the long allele and you're peer-reared, if anything, you have slightly higher than normal levels. But if you had a good mother, it doesn't make a damn bit of difference which allele you have. You have perfectly normal serotonin metabolism. Here's an example of a gene-by-environment interaction where arguably the nature of that interaction is attributable to buffering by good mothering. Good mothering is buffering those individuals, those offspring, carrying this allele from developing this kind of deficit. You see the same pattern when you look at aggression. Here, if you're peer-reared and you have the short allele, you have high levels of aggression, but same allele, good mother, your levels are as low or lower than individuals carrying the long allele in either rearing condition. It's even more dramatic when you look at alcohol consumption. Here, if you have the short allele and you're peer-reared, you drink to excess. But if you have that same short allele and you had a competent mother, you actually drink less than normal. What may be a genetic risk factor for individuals with poor early experience may actually be a genetic protective factor for individuals with good early experience. Gene-by-environment interaction, buffering by good mothering. And we see it throughout development. We run all of our monkeys through the infants born in the lab through a monkey version of the Brazelton Neonatal Assessment Scale. This monkey version translates almost perfectly item by item and cluster by cluster from the human version. And on one cluster in our monkeys, visual and auditory orienting monkeys who perform poorly on that during the first month of life tend to grow up to be impulsive and aggressive. The test is here. You see the test, move an object across the infant's visual field, see how well it can track the object. When you break the data down by rearing condition and genotype, once again, nursery rearing, short allele, poor performance on orienting, but same allele, good mother, normal orienting, normal development, etc. And it's not true. This pattern we see over and over again in other candidate genes. MAOA, rhesus monkeys have a functionally equivalent, structurally different, but a functionally equivalent polymorphism, as do humans. Caspi and Moffitt in their Dunedin study reported a gene-by-environment interaction involving MAOA, where humans with the less efficient version of this gene had higher levels of aggression, but only if they also had a history of child neglect or abuse. We've replicated that finding in our rhesus monkeys. We've replicated that same pattern now in 8 or 9 candidate genes: mu-opioid, neuropeptide Y, vdnf, dopamine transporter, DRD4. In all of these genes, we see the same general pattern. Here's SNPs and a CRH polymorphism, and the pattern is the less efficient version of the gene is associated with poor outcomes, in this case alcohol consumption, only in peer-reared monkeys. That same less efficient version of the gene in mother-reared monkeys shows either normal outcomes or better than normal outcomes. I think Jay may have something to say about this later on. So over and over for all these candidate genes, we see an impressive effect of good mothering. So we've been very interested in what constitutes good mothering and have spent the last couple of years really looking at it very, very closely. This is, for us, especially important because not only is good mothering seemingly protective of individuals who carry these risk alleles, but good mothering is passed on to the next generation. We now know by elegant studies by Dario Mastapiheri, by largely non-genetic means, so that mothers who have offspring who carry these so-called risk alleles, again so-called, can have normal offspring as a consequence if their maternal behavior is competent, and the females of their offspring will go on to be good mothers to their own offspring who may carry these so-called risk alleles. So here's a mechanism by which genetic so-called risk allele can stay in the population generation after generation with no apparent ill effect. On the other hand, if something happens to compromise the ability of that mother to take care of her kids, maybe there's been a death in the family. Maybe there's another family that's pushing them with, in terms of the family dominance. Maybe another troop has moved into the area that's competition. Maybe there's a food shortage. Maybe there's an increase in predation. Maybe there's a hurricane, climatic disaster like a Hurricane Katrina. Any one of those things that compromise the ability of a mother to take care of her kids will put those kids of hers who carry these alleles at risk for poor developmental outcomes. So let me give you some evidence, some examples of what we mean by good mothering. Here, this first video is a low-ranking mother who doesn't have very much clout in the troop. He's got a 2-month-old infant, and as the video starts, there's a fight breaking out to the right of the camera. And you can see the infant looking at that fight with some interest of going and checking it out, and watch what the mother does. So see the infant looking? So what does the mother do? She grabs the infant, grabs a flower, distracts the infant. And you can see the members of the fight going away. Look at the face-to-face interaction. That's not supposed to happen. Rhesus monkeys aren't supposed to do that. Only humans, maybe chimps. Okay, fight's over. Situation's clear. Off goes the infant. Mother's going to follow just to make sure everything's okay. If there were more time, I'd show it again. It's really a beautiful video. I've seen it 100 times. I see something new each time. It's a perfect example of a secure attachment. Okay, here's another infant. This one's 19 days old. You have to understand that 50 years of mother-infant research in rhesus monkeys, and nobody has ever reported face-to-face interaction. No one. Not Harlow, not Hine, not myself, but there it is. So once you start looking for it, you find that there's quite a bit of it. Blue is mother-infant interaction. Red is infant's interaction with anybody else. It's all mother-infant interaction. This is over the first 3 weeks of life, and then month 2, mutual gaze, lip smacking by the mother to the infant, lip smacking by the infant to the mother, back and forth. Why didn't people see it before? Because after 3 weeks, it's almost gone. It just drops out of the repertoire almost completely. What's happening at 3 or 4 weeks of age? That's when the infant starts leaving the mother to go and explore the environment. It's almost as if it doesn't need that face-to-face interaction to maintain the relationship anymore. It's already there. The infant knows it can go back to the mother if it gets in trouble. Now it's more important for it to be paying attention to other things in the world. And furthermore, if it stares everybody else in the eye, it'll get the crap kicked out of it, because that's the best way to start a fight among older monkeys. So it's probably important that this gets out of its repertory. But boy, during that first 3 weeks, here are the data. This is the lip smacking going down, and the amount of time the infant is staying, leaving its mother, spending away from its mother, is going up at the same time. But the other thing you can see is not only is it dropping out at 3 weeks, it's there from the beginning. It's there on the first day of life. So here's a 3-day-old infant, and that's not its mother, to the right. That's Pierre Ferrari. Pierre Ferrari is a junior scientist, brilliant junior scientist, from the lab of Giacomo Rizzolatti, the father of the discoverer of mirror neurons. We've heard a little bit about mirror neurons before in Parma. And the mirror neuron people in Parma wanted to know how early these things might be working. So they needed to see some infants. Who's got infants? Sumi's got some infants. So what you see is a still face by Pierre for 30 seconds. And the first thing you notice is that the infant's really interested in the face. Who said infants aren't interested in faces? Day 3. They're seeing each other for the first time, by the way. Now Pierre's going to open his mouth. Watch the infant. Ever heard of neonatal imitation? Tiffany Field, Andrew Metzoff 30 years ago. Newborn human infants can imitate the facial expressions of their mothers hours after birth. This sure looks like neonatal imitation to me. Here's another one. This is Ken Vaughn on the right, one of our texts. Another 3-day-old infant. Ken's going to stick his tongue out. Watch the infant. See it? See it? See it? Neonatal imitation. 3-day-old infant monkeys. Again, the thought of the Parma people is that this is mirror neuron. I'll play it again. The infant starts imitating after the 4th stimulus. The people in Parma think this is mirror neuron mediated and that it's there from birth. See it? So we've been spending the last couple of years making faces at baby monkeys, and we now know a great deal more about the phenomenon. First, as in the human case, it doesn't stay around very long. In humans, it's basically gone after a month. In rhesus monkeys, it's basically gone after a week, 4-to-1 ratio, right on schedule. Secondly, Metzoff and Tiffany Field reported that not every infant did this. In their case, they tossed those non-imitators out of the study because it was messing up their statistics. We're interested in those infants. So here, among nursery-reared infants, about 60% are imitators, about 40% are not. Interestingly, when we look at mother-reared infants on day 3, we get one shot at them. They are during their mother's postpartum exam when they have to be under mild anesthesia. We keep the infants on their mother's ventrum, but we can still make faces at them. And when we do that, 95% of the infants, mother-reared infants, imitate. So already by 3 days of age, you got a big rearing condition effect. So does it make a difference whether you imitate or not? Well, when we look at our Brazelton exams, the one measure, there's no difference. Red is imitators, gray is non-imitators, day 7, 14, 21, and 28. No difference in body weight or size. No difference in gross motor maturity, neonatal reflexes. But on one measure, the one measure on the Brazelton that looks at sensory motor integration, coordinated reach and grasp. Put a toy up there, the infant has to locate it, reach for it, and grasp it. Imitators do better significantly at each time point than do non-imitators. And when these monkeys get older and are at the age when they should be starting to play, those that imitated during the first week of life play twice as much as those who did not, whether it's month 4, 5, or 6, when play is really starting to take off. And when these monkeys get a little bit older, 2 years of age, and they've been living in stable social groups for 18 months, we find that those who failed to imitate in that first week have higher levels of self-directed behavior, sucking their thumbs, grasping their bodies, and they also develop spontaneous autistic-like stereotypies, whereas none of the imitators ever developed that. We just might have stumbled on a very early screen for risk for developing autistic-like stereotypies, at least in rhesus monkeys. I have some human colleagues studying autism in humans who are pretty interested in these findings. So what about, so where do you go from here? One of the things we're going to be doing is some interventions. We now know by 3 days of age whether you've got an imitator or a non-imitator. What happens when you start giving it as much visual facial stimuli as infants who are reared by their mothers are getting? Can you get them back on track? And what about the perhaps relationship with mirror neurons? Well, mirror neurons were discovered by Rizzolatti using single-unit recording from adult macaques strapped to chairs. There is no way I'm ever going to do that to any of my monkeys in the nursery. But you can look at EEG, and it turns out that down the road a piece from from NIH is the University of Maryland. We're one of the world's experts in human infant EEG works. Nathan Fox, a friend for decades. So we got Nathan involved, and we're now recording EEGs from infants during the imitation process. This was the very first one we did, primitive scalp electrodes, 3 of them only, but they pick up during imitation. We got enough signal, and we get different patterns at different stimulus conditions. We then move to the kind of caps that Nathan uses in human infants. This is our prototype cap. Kid's not very happy, cap doesn't fit very well, but we still get what appears to be desynchronization of low power mu rhythms at low powers in imitation conditions as opposed to none. Here's our most recent version of it. These are now pretty snazzy little caps, snap-on electrodes, imitation, and we find when we do that there is that same desynchronization of low wave, low energy mu rhythms. Here are the initial data, control periods at 6 kilohertz, high levels up here, stimulus of tongue protrusion, stimulus of lip smacking, actual imitation. There's your desynchronization. We now have an EEG signature. You don't see these patterns during non-imitation periods. You don't see these patterns in infants who fail to imitate. So unlike this, the imitation reflex that goes away after a week, we can follow this up developmentally, and that's what we will do. So our imitation, neonatal imitation work is going to continue for the foreseeable future. These are some of the people who were involved in this work, and these are some of the people who were involved in the largest study. I want to thank them for helping me out with this, and I thank you for your time.
Thank you very much. That was wonderful. One comment or question?
Yeah, one question. Have you ever tickled these young?
No, but we're going to start pretty soon. And I don't know what kind of chirp they'll get, but I think we'll get something.
Tickling in the future. Okay, one more.
Do you expect, you mentioned you won't mention this, but if you look at animals when they get older, you said many of the behavioral differences between period and mother-reareds disappear?
Yes, we can by putting them in nice, I would call them therapeutic environments, just really stable, supportive social situations. We can turn around the behavior. We can turn around the HPA axis stuff. We make the at-risk mothers their mothers, they show normal mothering behavior, and their kids are indistinguishable from the offspring of mother-reared infants. And of course, the story is, can you turn the genes back on too?
So that's the question about methylation.
Then that's what we're going to look at next as these guys grow up.
Yeah, great.
Thank you.
Steve, may I ask a question? Here.
Last one.
You speak of gene-environmental interaction as if it's a special condition of gene-environmental studies, when we know that the environment controls and regulates gene expression.
Yeah,
genes do not
it's one of the astonishing findings of the last 5 years. Most astonishing to me. Gene expression.
But we've known this for more than 5 years.
Yeah, but nobody believed it. Now we've got the data to show it. We, meaning it and others. And so what it comes down to is gene expression, from my way of thinking, is as plastic as anything else.
Exactly, and it's all gene-environmental interaction. There's no such thing as gene expression as a pure factor in development. And we have, I think, been misguided in terms of the kinds of research and the Genome Project and so forth, that it's all genes. And we forget that there's an environment involved, and that the environment is really the trump card in all of this. And so if you had to choose between the gene path and the environmental path for constructing conditions to produce a happy child, which path would you pursue? This gene emphasis, unfortunately, is producing a process that feeds into the pharmaceutical industry in creating drugs and more pills to correct a metabolic disorder, when all you have to do is look at Mother Nature, as you've done.
At the risk of getting myself in trouble with my thing, I would like to point out in the last 2 to 3 years, and especially in the last year, there's been a major shift in emphasis from the very top at NIH to really start taking environments seriously, how we measure them, and more importantly, to better understand these, what they now call gene-environment interplay. And it's been a long time coming as far as I'm concerned, and I see nothing but good news coming out of it as a result, much different from the past.
I've not yet seen that, and that is a good news.
Thank you. Thanks very much.